behalf of Excel College of Pharmacy, Kumarpalayam, I warmly welcome you all to the International Expert Talk Series, Day 6, organized by the Department of Pharmacy, Practice and Pharmacology. We are honored to have all distinguished participants, faculty members, research scholars, and students joining us today. At the outset, I would like to extend my heartfelt gratitude to our management, principal, and organizing committee for providing us with this great learning platform. Today's expert session will enlighten us on network pharmacology based on prediction of therapeutic target drugs in diabetic mellitus, a highly relevant and impactful area in pharmaceutical research. It is my privilege to introduce today's eminent speaker, Dr. Sasikala Chinnappan, Madam, Assistant Professor, Head, Pharmaceutical Biology, Faculty of Pharmaceutical Sciences, UCSI University, Kuala Lumpur, Malaysia. She holds her B.Pharm from Dr. MGR University, M.Pharm in Pharmacology from Shastra University, and PhD in Pharmaceutical Sciences from Jawaharlal Nehru Technological University, Kakinada. With more than a decade of teaching and research experience, Dr. Sasikala has contributed immensely in areas like natural products, diabetes mellitus, wound healing, nephrolithiasis, toxicity studies, and innovative drug delivery approaches. She has published widely in reputed international journals, reviewed scientific manuscripts, and guided numerous scholars. We are truly honored to have her with us today. Without any delay, I invite Dr. Sasikala Chinnappan, Madam, to kindly deliver her expert talk. Ma'am, the virtual stage is yours, please.
Okay, thank you very much, sir. Good morning, everyone. First of all, I would like to thank Excel College of Pharmacy and convener and organizing committee. And also, I would like to say my special thanks to Pragasam sir as well. I hope you can see my screen, okay, my PowerPoint, right?
Yes, ma'am.
Okay. So I'm Dr. Sasikala. As a head, I'm working as an assistant professor and Department of Pharmaceutical Biology in UCSI University, which is in Kuala Lumpur, Malaysia. So today, I would like to talk about network pharmacology-based predictions of therapeutic target in diabetic mellitus. So as we know, everyone, drug is any chemical substance. When we introduce into our body, it produces some biological effect, right? So that substance can use to diagnose or prevent or treat or cure diseases. Okay, so most of us have taken a drug at some point in our life. For example, paracetamol we are taking to reduce the fever or pain. Or some people, they are taking insulin to reduce the diabetic condition. So behind every pill, there is a long journey of science. There are many steps actually the drugs go through. There are 5 drug development phases we can observe. It starts with the discovery and development. For example, we are finding a lead compound. Okay, so that lead compound maybe from natural source or maybe synthetic source, right? Or sometimes we are using computer-based drug design to identify lead compound. So once we found the lead compound, what we are doing, we are moving to the preclinical research. So during preclinical research, we are doing toxicity study and also we are doing some pharmacological assay. Okay. So when you see toxicity study, we are doing acute toxicity, subacute toxicity, and chronic toxicity study. Why we are doing? To identify the toxicity profile of your lead compound. Okay, so because first we want to know whether our lead compound shows any toxic or not. Okay, so after that, we are moving to the pharmacological assay to find out the efficacy of our drug. Okay, because if it doesn't have any efficacy, there is no point of moving to the next development stage. So your lead compound shows some biological activity. Okay, it shows some efficacy. Then we are moving to the clinical research. Okay, so during clinical research, what we are doing? Okay, we are selecting human. Okay, there are different phases of clinical research. Phase 1, we are checking our lead compound on healthy volunteers. Okay, phase 2 and phase 3, we are selecting disease patients. Okay, so this development stage also we are confirmed about our efficacy and safety of our lead compound on human. Okay. So once everything is okay, we are submitting all the data to the FDA or other regulatory authorities, okay, to get the approval. Okay, now if you get approval, then we are market our drugs, right? So, but sometimes, still we need to monitor the drugs, okay, safety monitoring in large group of population after post marketing also. Okay, so this is the different drug development stages we can see each and every field. Okay, so now you can see, as I said, okay, so for example, 1,000 molecule, if you are finding your lead compounds are 1,000, 10,000 lead molecule, okay, but how many it is coming to the preclinical stage? Only 200, only it's coming to preclinical stage. Okay, so the remaining drug molecules are failed in the character. Okay, so from the 200, how many is coming to the clinical? Only 5 molecules are coming to the clinical research. Okay, so now here also we are checking safety and efficacy of the drugs on human. So finally, when it is reached, it reached 1 or 2 molecule. Okay, so we are starting from 10,000 molecule, but is end up with the only 1 molecule. Also, it is not easy. Okay, so how long we are taking? More than 10 years we are taking to develop new drugs. Okay, it's a very long journey of discovery. Okay, so we start with the lovastatin. Okay, so we all know lovastatin we are using to reduce the cholesterol level. Okay, so in 1960, the heart disease was the world leading killer disease. Okay, so we couldn't find any safer drugs to treat the heart disease. Okay, so after that, what happened? Okay, some scientist, Endo scientist, is a Japanese biochemistry scientist. He found that, okay, so one drug from, okay, fungus, okay, he was checking with the cholesterol. He thought that actually it can able to reduce the cholesterol level. Okay, but what happens during the toxicity study? Okay, it was failed. Okay, because these drugs like simvastatin actually to produce some of the kidney disease and skeletal muscle disorder. Okay, so the simvastatin found by the endo was failed during the time. Okay, so later Merck scientists found another drug that is a lovastatin. This also is actually found from the fungus. He was find that actually this drug was very good, high efficacy toward the cholesterol reductions with minimal side effect. Okay, so when you see the journey, actually they started to develop their drugs in 70s, but the approval of drug is at 80s. So how long they are taking? More than 10 years they took to develop a single drug. Okay, so nowadays we are talking about the AI, right? So any field if you take, there is actually nowadays we are looking into the AI. Okay, so do you think that AI also can involve in the drug development? It can help in the drug development? Yes, of course. Okay, so when you see in whether is the lead development of any compound or preclinical, clinical, all the phases the AI can help. Okay, so for example, clinical trial. How can help in clinical trial? This AI can help in identify the suitable patients, and also it can able to help in the enrollment. Okay, so also it can able to monitor the overall process of clinical trial. Okay, it can take the data and it can automatically store the data. Okay, so next is the target identifications and validation. Okay, if you have lead compound, but you don't know what is the target of the lead compound, you don't know what is the therapeutic use of lead compound, then this AI algorithm can use to develop a new protein target or gene target. It can identify your lead compound therapeutic use. Okay, and next we need to know whenever we talk about the pharmacology, the main thing is a pharmacokinetic parameter also. Okay, so the AI can able to predict the drug property. Okay, so for example, if your lead compound you don't know about what is the absorption distribution. Okay, so before go to the actual experiment, these AI can able to predict what is the ADME character of your drugs. Okay. And next you can see, okay, so this AI algorithm also can able to predict what is the dose, what dose it is need for the patient. Okay, so that predictions also can given by the AI. Okay, so next is the preclinical testing. Okay, preclinical testing, you actually, before you go to the in vitro, in vivo study, okay, some of the database also can help in actually to check the what is the preclinical activity of your lead compound. Okay, so next is the process of genomic data. Okay, so a algorithm can able to give the okay, so genetic data which is related to the what lead compound can act on which type of gene. These algorithm also can help to detect the gene. We can make a personalized medicine by this AI tool. Okay, so finally, okay, so adverse effect, because each and every drug have some own adverse effect, right? Okay, so before actually we do with the toxicity study on animal, this AI also can able to give the predictions on what is the adverse effect of your compound. Okay, so now, so I want to compare the traditional drug development and network pharmacology, that is AI assisted drug discovery. As I said, when we see traditionally what we are practicing, okay, so we are more than 14 years actually is we are spending time to develop a drugs. Okay, so from the lead identification of the lead compound into the and FDA approval. Okay, so the cost, when you see the cost, okay, cost also is high. So more than $3 billion of dollar we are spending to develop a single compound. Okay, so that's why we are thinking how we can reduce the cost, or how we can reduce the timing also. That is the biggest challenges in the drug development. Okay, so this AI assisted learning, okay, so can definitely will be reduce the time frame. Okay, so from 14 years to 7 years, half can reduce. Okay, like the cost wise also it can reduce to 50 to 60%. That is the beauty of AI. Okay, so now we can see how the pharmacology is helping here. Pharmacology is a new approach, because nowadays, many people, they are talking about the network pharmacology, right? So before we are going to the actual experiment, okay, so we are using network pharmacology to identify what is the preliminary data about your lead compound. Okay, so this is actually really help in the drug design. Okay, what is network pharmacology? Is a system biology. It means we are taking whole system as a one. Okay, then we are construct the network. We identify your lead compound act on how many target. Okay, so next, by network pharmacology, we can analyze the what is the side effect. Okay, so also what is the new target, what target area, what is the receptor, what is the gene which is involved in the disease pathway. So these all actually can predict by network pharmacology. Okay, so that's why it's actually is helping in the drug development in many medicinal research and traditional research in network pharmacology. Okay, for example. Okay, so one of the complicated disease is the cancer, right? So if you want to find out the molecular mechanism or activity of anti-cancer drug, we need to spend more time, and also we need to spend money also. Okay, so sometimes we will get results, sometimes we won't get result. That is, sometimes it's a waste of money, waste of time also. So that's why here you can see that when we imply that network pharmacology, actually we can able to predict. Okay, so what may be the molecular pathway. Okay, so after that, if you go to the experimental design, so we can reduce the cost and time also. Okay, so now you can see here, mostly what we think whenever we find a lead compound, we think that actually, okay, one drugs will be act on one receptor, it produces some biological effect, right? Okay, like paracetamol you take. So paracetamol, it can give to reduce the fever. Okay, But what is the off target? Off target is like a disease side effect. Okay. So apart from therapeutic effect of paracetamol, sometimes paracetamol can give the liver toxicity, right? So this is actually traditionally what we believe. But the network pharmacology, what it says, okay, so a single drugs can able to target on multiple protein, okay. So multiple way it can able to control the disease, okay. So it can give multiple therapeutic use, okay. So that's why really we are looking into the network pharmacology, okay. So to find out more therapeutic use of a single compound, okay. So now you can see here is how it is developed. Who developed this one, network pharmacology? Okay, it's actually started in 1999. Okay, started with actually Andrew Hopkins. Okay, he was thinking about the network pharmacology. He designed the network pharmacology. Okay, so slowly it developed in 2000. Okay, so they come up with some of the database like drug bank. Okay, so we can use the KEGG genome database, omic database. Okay, so these are actually introduced in 2000. Next, 2010, actually you can see here, they use the network pharmacology in traditional medicine, okay, traditional Chinese medicine. So now they say that once we know what is the therapeutic use of your traditional medicine, then this traditional Chinese medicine, they develop the formula, okay, herbal formula, Chinese herbal formula, they develop, okay. So by using network pharmacology, okay, and then later in 2010 until 2020, okay, so they actually want to implement this one into some complicated disease like a cancer, diabetic, and some of the neurodegenerative disease, okay, by using cytoscape, okay. So they construct what is the molecular pathway in involvement in the different type of complicated disease, okay. So nowadays is many people that are using network pharmacology, okay. So we are trying to use the olden drugs, okay. So what is the new therapeutic target, okay, drug repurposing. So that's our concern now, okay. For example, if we once we believed is a single pathway is caused diabetic mellitus, but the approach is failed because we can't say only one pathway which is involved in diabetic mellitus. There are many pathways are involving, okay, so to cause the diabetic mellitus. So we can able to target in different way to control the diabetic mellitus, right? Okay, so that approach actually we can see in the network pharmacology. Okay, so you can see here, if you want to work network pharmacology on herbs, okay, it's or we can take any old drugs, okay, repurposing also you can use. You can identify the new target of the old drugs, okay. If some people they want to see how it's work on traditional medicine like Ayurveda, Siddha, Chinese medicine. Okay, so for example, if you take herbs, it contain rutin, quercetin, many phytochemical we can see, right? Okay, if you take rutin can able to reduce the TLR4, toll-like factor. Okay, so which is responsible for inflammation. Okay, so other phytochemical quercetin is actually involved to reduce the NF kappa, which is involved in the cancer. Okay, so the beauty of network pharmacology here, the quercetin is not only reduce the NF kappa pathway, also you can see this is act on the TLR4 also. It means the quercetin is not only effective in cancer, the quercetin also can reduce the TLR4, can reduce the inflammation. Okay, we can observe more than one reductions of therapeutic use of the phytochemical. Okay, so this one we can able to identify by using network pharmacology. Okay, so now here I want to discuss about what is applications, mainly where we can use the network pharmacology. As I said, is a drug discovery. Okay. So discovery of the new compound, multi-target, single compound, it is act on the multi-target, it gives the multiple therapeutic use. Okay. So next we can see is a drug repurposing. We are taking old drugs. Okay, we are finding a new therapeutic use of the old compound. That is the main applications. Okay. So now when you see these main applications in terms of traditional medicine, the first thing is actually network pharmacology. First is give the evidence-based uses. You have some evidence by doing network pharmacology. Okay, by using that evidence, we go to the next level. Okay, so you can design the formula, herbal formula for some of the therapeutic uses. Maybe you can target on asthma, you can target on diabetic mellitus. Okay, so also this network pharmacology can give what is the molecular mechanism, a mechanism of action of your formula. Okay, your herbal formula. Okay, so next, in terms of pharmacology, if you see, this network pharmacology can give the, okay, so to develop a new compound, and also it says what is the molecular mechanism of your compound, what is the side effect of your compound, is there any drug-drug interaction of your compound, whether we can go for drug repurposing of new therapeutic or not. Okay. So in terms of drug research, what it gives, how can help in the research? As I said, we can find a new novel compound. Okay, so we are going with the in silico study. Okay, so we are going with the network pharmacology. So what it do, it can reduce the timing and also it reduce the cost. Okay, and next, definitely giving the signaling, can determine what is the signaling pathway which is involved in this particular disease. Okay, so and next we can identify what genes are involved in this particular disease, and we can say which is in diagnostic marker of the particular disease or not. Okay, so also we can see your lead compound is only act on the normal bacteria or virus or resistant bacteria or virus. This also we can able to predict by using network pharmacology. Okay, so this is an overall picture of the network pharmacology. You can see here, network pharmacology is giving some idea only. Okay, so we cannot say is 100% we can use this data to go for any clinical trial. Okay, so not like that. Okay, so network pharmacology, once you design here is the network constructions of your compound with the disease, what are you supposed to do? We need to go for the in silico study. We need to have some validations before go to the clinical trial. Okay, so in silico study is a computerized program. We are seeing that what is the ligand and receptor interactions. And next we need to go with the in vitro study. First we need to check whether it shows any actions or any efficacy or not of your lead compound. Then only we are moving to the in vivo, right? Okay, so after network constructions, you compulsory need to do some validation of your lead compound before go to the clinical trial. Okay, so now you can see here, actually we try to use this network pharmacology in different diseases. We are working with Alzheimer's disease, Parkinson, and diabetic disease. Today, I would like to focus on diabetic mellitus. Okay, so we all know that many people are nowadays are suffering from diabetic mellitus. Almost 589 million people are worldwide. They are living with the diabetics. In India, 10.5% of Indian adults they are living with the diabetics. Okay, in Malaysia, 443K people are living with the diabetic condition. So that's why there is an urgency to find new therapeutic use of the new drug development toward the this type of complicated disease. Okay, so we have selected alpha mangosteen. Okay, so alpha mangosteen, you all heard that is a natural xanthone derivative. Okay, so it's isolated from the pericarp of Garcinia mangostina. Okay, is a fruit. Okay, so you know about that fruit, right? Okay, so mainly as per literature, you see that alpha mangosteens are very potent in anti-cancer property, which can induce the apoptosis of cancer cell, inhibit the proliferation of cancer cell. Okay, so it is act against the breast and prostate and colon cancer. Okay, so in diabetic conditions, it can improve the insulin sensitivity. Okay, so also it can decrease the alpha glucosidase enzyme, which can help to manage the hyperglycemic conditions. Okay, so next, anti-inflammatory property, it can suppress some of the inflammatory marker. Okay, NF-kappa pathway can reduce cytokinins, it can reduce by the alpha mangosteen. Also, it can use to work against the bacteria, fungi, some parasites. Also, is a neuroprotective actions we can use for the Alzheimer's disease. Okay, so now you can see here is a general workflow whenever we do the network pharmacology. Okay, first we need to define what is the disease. Okay, you need to define what is your lead molecule. Okay, so we have selected disease is a diabetic, we selected. Drug we have selected alpha mangosteen. Okay, so the next step we need to collect the the molecule from the pumpkin smile, and we put that smile into the Swiss target predictions. Okay, so this is Swiss target prediction is another database. Okay, so need to find, okay, so what are the target gene, okay, by alpha mangosteen. Then next we collect genes which is involved in diabetic mellitus from the database, gene card database. After that, we run bioinformatics tool and find Venn diagram to identify what is the common gene between disease target gene and lead compound target gene. Okay, need to find what is the common gene between your lead compound and disease. After that, we use cytoscape to construct the network. We use the cytohubba. We find the top 10 gene which is involved in this pathway, diabetic pathway. Okay. Finally, we use David database to identify what is the molecular pathway behind this. Okay. So let's see one by one. Okay, now you can see this picture. This is called as the network construction. What you can observe here, you can observe is a node. Okay, node is for example, you consider disease is a node. This all is called as the edge. Okay, the green color, purple color or edge. What it says here, what this disease are related to what gene. Okay, so what are the genes are involved in this disease? It can shows that what are the genes which is involved in the herbs, this phytochemical. These constructions we can create by this graphical map, we can create by network pharmacology construction. Okay, so now you can see here. Okay, this is the data we observe from the Swiss ADME. Okay, so because we want to know what is the pharmacokinetic profile of the alpha mangosteen. This is actually always a prediction. Okay, so from here you can see that the alpha mangosteen shows high GA absorptions, and there is no any blood-brain barrier cross. Okay, so it's very safe. Okay, and next it can shows the glycoprotein substrate. No, it means it is not effluxed by the glycoproteins. It can diffuse into the cell membrane. Okay, also you can see here there is no inhibitions of cytochrome except one cytochrome you can see, that is the cytochrome 2C9. So what actually from this, what we can understand, okay, it is like safe drug, but if any drugs are metabolized by cytochrome 2C9, we can't take with the alpha mangosteen like a warfarin, okay? Because it's inhibitor, it inhibits the metabolism of warfarin. So the warfarin side effect you can observe. So this A algorithm predict what is the pharmacokinetic profile of the alpha mangosteen, and also you can see the toxicity profile of alpha mangosteen. Okay, is you can observe here it is inactive in some of the organ. Okay, so like respirations, okay, hepatotoxicity, neurotoxicities are inactive. Also, it predicts that there is no any carcinogen, there is no any cytotoxicity effect, but it predicts it alpha mangosteens can cause the nephrotoxicity. Okay, and next, this is you can observe in Swiss ADME platform. Okay, this is called as the boiled egg plot. This picture shows, okay, so whether your drug is cross the GI or it can cross the blood-brain barrier. The white color is represent the GI. The yellow color represent is the egg yolk. Okay, like egg yolk, look like egg yolk. It means represent it cross the blood-brain barrier. So here you can see a molecule. Okay, this is alpha mangosteen, which can cross the blood-brain barrier. You can see it's only cross the white color only, not cross the yellow color. It means it cross the GI, but it doesn't cross the blood-brain barrier as per this AI prediction. Okay. So now this is a network. Okay, you can see in the middle is the alpha mangosteen. This will be constructed by 100 target protein. Okay, there are 100 protein you can see surround the alpha mangosteen, which means these constructions we got from the cytoscape. Okay, so next you can observe here, as I said, okay, so total 100 molecules are targeted by your lead compound. Okay, so disease genes are almost 18,245 genes it's a disease genes we obtain from the gene database. Okay, so by using bioinformatic tools, we see that what is the common gene between target of your lead compound and disease. Okay, so now you can observe here, totally 93 genes are common genes. Okay, between your lead compound and disease compound. So these 93 genes, we move to the next level. Okay, you can observe here there is some constructions you can see. Okay, so this is actually done by string database. What string database will give? String database will give the protein-protein interactions. It means interaction between two protein. Okay. So by using this data, okay, the protein-protein interaction, which one is the high protein-protein interactions we collected. Okay, so then we move to this data, we put it into the cytoscape. Okay, so we construct the network of the gene, which is obtained from the string database. Okay, so now you can see here, when this was actually is done in the Sato Hubba. Okay, so this is a part of cytoscape software. Why means, as I said, there are more than 92 compound actually we found, 92 genes we found, okay, which is involved in the disease. But from the 92, okay, we want to select what is the top 10 gene which is involved in the diabetic. Okay, because we want to see which is more significant genes involved in the diabetic mellitus. I want to work on that. Okay, so by seeing this picture, you can see top 10 genes. The blue color is the MAPK pathway. Okay, it's a central hub gene. And next you can see the yellow color mTOR, MAPK, TNF. Okay, so this is the another pathway. And red color, you can see AKT, HSP, and TCNN. Okay. These all top 10 gene actually can involve in the diabetic. Okay. But according to the score, okay, you can see score of the top 10 gene. Okay. So here you see which is the highest score. The CTNN 54 is the highest score. Okay, catenin beta. Okay, so all the 10 genes are which is involved in diabetic mellitus, but the highest score, highest probability, what we can see, the 54, the highest score we consider. Okay, as I said, this is a network constructions. We can, if you want to work on 10 gene, we can work on 10 gene also. You can identify, you can go to the next level. Okay. So here I have here the given actually top 10 genes which is involved in the diabetic mellitus as per construction, network construction. Okay. So as I said that after we construct the network pharmacology, we move to the validation process. The first validation process is a docking study, in silico study. Okay, so this is actually done by the CBDA. Okay, so one of the platform to identify to do the docking study is the CBDA. Okay, when see the comparison between top 10 gene, which is the highest scoring binding energy scoring you can observe is minus 10.4. That is the mTOR pathway. Okay, so you can see that CTNN, as per the network construction, CTNN V1 is the highest score. But though, okay, so when we are coming to the in silico study, which is the highest energy, you can see highest interactions with the ligand, you can see is the mTOR protein. Okay, so that's why they said we need the validations after we are doing the network pharmacology. Okay, so now you can see here. Okay, so this is a ShinyGo. This is another platform, is a ShinyGo. We want to see how the mTOR pathway, because just now we told that the mTOR pathway is the highest binding energy you can see. So this mTOR pathway, you can observe it is involved in many of the diseases. Okay, since we are talking about the diabetic mellitus, you can observe here the mTOR signaling pathways are involving in the insulin resistance and also which is involved in diabetic complications. Okay, like diabetic nephropathy, neuropathy, and okay, so also these mTOR pathway always we talked during the anti-cancer drug therapy also. Okay, so from these pictures we can say that yes, alpha-aminosysteins actually is linked into the diabetic mellitus and also in cancer. Okay, so you can see here, this is called as the gene ontology analysis. Okay, why we want to do a gene ontology? This is like by using David database, we done this one. Okay, so you can see we are comparing with the enrichment score. If it shows that what is the probability, is it whether it is significant or not. If it is, the bar is high, which is mean it is is more significant. That pathway is more significant when compared with the other pathway. Okay, this picture says that one. Okay, so this graph, okay, so can shows that what is the biological process, how the alpha-mangosteen which is involved in biological process, cellular component, and molecular functions. Okay, so in the biological process, you can observe which is related to this PPA gamma signaling pathway, which is involved in diabetic, which can increase the uptake of the glucose. When you come into the cellular level, you can see NF-kappa pathway here, because when we reduce the NF-kappa pathway inflammations in pancreas, we can increase the insulin secretions. And also at molecular level, when you see it is highly involved in the serine enzyme kinase activity, this enzyme activity is also which is help in insulin production. Okay, so this is actually, you can observe, this is a fold enrichment analysis. What is fold enrichment analysis here? Okay, so we see that what are different disease pathway. Okay, so with a significantly different between the disease. Okay, so if you have the bubble is big, it means it's more significant different. Okay, so more significantly differ from other pathway. So this is actually is involved in the cancer pathway. As I said before, also you can see is also the bubble is small, but though it is involved in the insulin resistant. Okay, this is called as the fold enrichment data. Okay, as I said here, when you do docking, you can see there is the interactions between our alpha mangosteens and different protein, serine protein, asparagine proteins interactions with our compound. Okay, so next we want to talk about the mTOR pathway, because as I said, the mTOR pathway is highly shows the binding energy. This is called as the mechanistic mammalian target of rapamycin, is a kinase. Okay, so under this we can see mTOR C1, okay, and C2. There are two proteins. This is a complex protein under mTOR. What actually is the function of these two? How we can link to the diabetic mellitus? mTOR C1, which can regulate the cell growth of the pancreas, and also which can increase the uptake of glucose, okay, in peripheral tissues, and also which is involved in the lipid metabolism. When you see mTOR C2, another pathway, another protein, which can regulate the cell survival of the pancreas, cytoskeleton, and insulin secretion. So both complex, both protein which can work together to maintain the level of glucose in our body. Okay. So what actually molecular pathway, whenever any growth factors act on the insulin growth factor receptor, which can initiate, which can start the signaling of PI3 AKG. Okay, so we all know about that. So this AKG pathway, which can produce the mTOR protein. Okay, as I said, there are two mTOR protein. So mTOR C1, which can increase the uptake, mTOR C2, which can regulate the cell growth of the pancreas, which can help in the insulin secretion. But if anything happen, okay, so if it is overproduction of this protein, lesser production of this protein, both can able to causes the insulin resistance. Okay, so it can impair the glucose uptake. Okay, so it can disturb the production of the insulin. That's why it can produce diabetic related complications of diabetic mellitus. Okay, so you can see this is the pathway. As I said, whenever the growth factor is activated, PI3K, AKT will be activated. Okay, so which is activate the mTOR pathway. Okay, so here you can see the regulations. We can, if it is mTOR pathway will be inhibit. Okay, so totally it will be total actions of the protein will be reduced. So that also may be probability of produce the diabetic mellitus. Okay, it causes the insulin resistance. Just now I told. We want like a regulator, because overproduction, underproductions both can cause the insulin resistance. So we need to find a way to how to regulate. If it is high mean we want to reduce, if it less means we want to increase. So we need the regulator. Okay, so we need to always maintain the optimal level of the mTOR protein. Okay, so that way we can able to help in the management of diabetic mellitus. Okay, as I said that if it is you want to develop a lead compound which can act on directly on the mTOR, Okay, so what will be happen? There is a complete blockage of the mTOR protein, production of mTOR protein. As I said, it also complete blockage also can lead to the insulin resistance. So we don't want that. So how we can able to regulate? Maybe we can think about the allosteric binding. Okay, so you all know that allosteric any lead compounds are bind with the allosteric area. So is a non-competitive binding site which can able to regulate the actual binding site. So which can help to regulate the signaling pathway. Okay. So when you see a limitation of the network pharmacology, sometimes database, sometimes the database is outdated. Also sufficient information we won't get. Okay, so always we need to confirm this data with some our literature. Okay, so literature in terms of we can take from PubMed or Science Direct. Okay, Google Scholar, we can use and we can able to compare. Okay, so protein interaction is new. Okay, most powerful tool. Okay, so but always we cannot able to rely on that. Okay, so as I said, we need to go with the evidence-based experiment to confirm the network pharmacology. Okay, so these network pharmacologies we can't stand alone. Okay, in the drug discovery development, we need to go with the experimental design to find out what is the network pharmacology. Okay, so how we can supplement or complement your research. Okay, that way we can able to use the network pharmacology. Okay, so network pharmacology is an integrative system biology. It's a computerized prediction biology. Okay, so everyone we need to know how we can use the AI or network pharmacology in some of the complicated disease like polypharmacy management in diabetics. Okay, we need to identify, as a pharmacist, we need to identify what is the pathway which is involved in the diabetics or other complicated disease. Okay, so we need to train the pharmacist using computational approach, okay, to design or to understand a new drug target. Okay, so thank you all. If you have any questions, then you can ask me.
Thank you so much, ma'am, for the insightful and inspiring session. We are now open for a brief question and answer session. Participants have posted their questions in chat box, ma'am.
Okay, we'll see.
Ma'am, what do you feel are the biggest challenges researchers will face in applying network pharmacology to clinical practice?
Okay, so they're asking actually what is the biggest challenges researcher will face in applying network pharmacology to clinical practice. Okay, yes, so as I said, actually, we can't say that challenges. This is in terms of we need to take as a positive way only. Okay, so in clinical trial, whether we are taking patient recruitment, okay, so are to compile the data to identify which genomes are which type of data actually the patients for information. For example, you already designed that. Okay, so you need the patients with this category. You have some inclusion criteria, right? Okay, so when you use the AI database, this algorithm will be easily detect whether these patients are actually is correct for your clinical studies or not. We need to see the positive way only. Okay, so I think we need to implement. After implement of network pharmacology only, we can say that is any challenges. My opinion actually, this will help the clinical practice, not in negative way. I think we won't feel any challenges here. We need to find a way how we can implement the network pharmacology.
Okay, ma'am. Okay, ma'am. Next question. Could you brief the recent advancement in drug repurposing that you come across in your research?
Yes, actually, we are working on that actually, but the recently we are talking about the metformin. Metformin is actually you are using for diabetic mellitus, right? But
Yes, ma'am.
nowadays we are using repurposing in the development stages or in the phase 1. Actually, whether we can use this drug for as an anti-cancer drug. Okay, and also dextromethasone during COVID time. Okay, so that is actually by using network pharmacology only we designed that dextromethasone is a well controlled in respiratory distress during the COVID condition.
Ma'am, next one, why is that diabetes mellitus is suitable disease model for network pharmacology research?
Oh, sorry. Actually, it is. Hi, Aruna. I want to say that actually this is not only in diabetic mellitus. As I said, it is not. But I'm working today. I presented on diabetic mellitus. It is not only diabetic mellitus. We can implement at any disease. Okay. Recently, my paper is accepted. I was working in network pharmacology on Alzheimer's disease and Parkinson disease. Okay, so if anyone you have any idea with the other disease with the network pharmacology, maybe you can approach me. I can guide. No issues on that.
Okay, ma'am. Any participants have questions, you can unmute and ask, please. Okay, no one. Let's conclude the session, ma'am.
Okay.
On behalf of Excel College of Pharmacy, I sincerely thank your esteemed speaker, Dr. Sasikala Chinnappan, madam, for sharing her valuable knowledge and expertise and clear presentation have truly enlightened our students, research scholars, and faculty members. A special thanks to our convener, Dr. R. Manivanan, sir, and organizing secretaries, Dr. M. Rangapriya, ma'am, and Mrs. M. Aruna, ma'am, for their constant efforts. Finally, heartfelt thanks to all participants for making the session interactive and successful. We look forward to meeting you all again in the next session of the International Expert Talk series. Have a great day ahead. Thank you, ma'am.
Okay, thank you very much, sir. Okay,
Thank you.
thank you all.